Hi, welcome to Topper Learning. I am Himangi. We will be studying together science, the subject of theories, principles and facts. Today, we will begin with one of the term 1 unit, the world of the living. This unit carries 29 marks. We will begin with the chapter life processes. We all perform different functions every day. We work, we play, study every day, talk to our friends, walk, eat and many more tasks. What makes us capable of performing these tasks? How our body does not give up on doing these tasks? Do you know its answer? Yes, correct. It is because of life processes. The definition of life processes is the processes which together perform maintenance function of living organisms. I repeat, the processes which together perform maintenance function of living organisms are called life processes. Now, what is the significance of life processes? The life processes prevent the damage and breakdown of the normal body functions. There are 4 important life processes: nutrition, respiration, transport and excretion. Today, we will be learning about nutrition as a life process. Today, we will cover the definition of nutrition, modes of nutrition, that is autotrophic nutrition and heterotrophic nutrition. Under autotrophic nutrition, we will cover the structure of chloroplast, structure of stomata and the mechanism of photosynthesis. Under heterotrophic nutrition, we are going to cover the 3 types of heterotrophic nutrition. Their examples: nutrition in amoeba and nutrition in paramecium. At the end of the class, we will also practice answering some important questions. Let's begin with the introduction to nutrition. Now, do we all need energy to perform tasks? Yes. Similarly, all life processes also need energy. Now, from where do we get this energy? We get this energy from food. The process of obtaining and consuming food is called nutrition. Every organism has its way of obtaining and consuming food. Based on this, there are 2 major modes of nutrition: autotrophic nutrition and heterotrophic nutrition. Now, let's first see what autotrophic nutrition is. In autotrophic nutrition, organisms synthesize their own food. Now, to synthesize their food, they utilize simple inorganic substances. So, autotrophic nutrition can be defined as it is the mode of nutrition in which organisms synthesize their own food from simple inorganic substances. Organisms which show autotrophic nutrition are called autotrophs. Again, organisms which show autotrophic nutrition are called autotrophs. Remember, in the science subject, definitions are very important from exam perspective. Green plants, green algae and some bacteria are the examples of autotrophs. On the slide, you can see the pictures of plant. Then there is another image of a green alga called volvox, and there is a colony of photosynthetic bacteria. They are called cyanobacteria. Now, let's further learn how plants synthesize their food. It is by the process photosynthesis. Let's define photosynthesis. Photosynthesis is a physiological process by which plant cells containing chlorophyll produce food in the form of carbohydrates by using carbon dioxide, water and light energy. Oxygen is released as a byproduct. The definition of photosynthesis is very important. Hence, I am repeating the definition for you again. It is a physiological process by which plant cells containing chlorophyll produce food in the form of carbohydrates by using carbon dioxide, water and light energy. Oxygen is released as a byproduct. You can see the chemical equation on the screen. 6 molecules of carbon dioxide and 12 molecules of water in the presence of light energy and chlorophyll produce 1 molecule of glucose, which is a carbohydrate, 6 molecules of water and 6 molecules of oxygen. Here again, since oxygen is released as a byproduct, you will see an upward arrow next to the oxygen. Remember, for every one photosynthetic cycle, 6 molecules of carbon dioxide will be converted into 1 molecule of glucose. Now let's see how much you have understood so far. Question is, complete the reaction by replacing A, B and C. So, if I replace A by 12H2O, B with C6H12O6, which is glucose, and C with 6O2, I get the complete equation for photosynthesis. I hope you all too got it correct. The raw materials required for photosynthesis are chlorophyll, carbon dioxide, water and light energy. Each has an important role to play during photosynthesis. Let's understand how these raw materials are obtained by the plants, for which we also need to know some structures. Let's see. Chlorophyll. It is a green pigment present in chloroplasts. Now let's understand the chloroplast. Chloroplast is a cell organelle found only in the plant cells. It is an oval shaped organelle. You can see the diagram of the section of the chloroplast on the screen. You will see there are 2 membranes which cover the chloroplast, an outer membrane and an inner membrane. In the cavity of the chloroplast, it has a colorless ground substance called stroma. In the stroma, if you observe the diagram, in the stroma you will see many flattened sacs are present. These sacs are called thylakoids. Now chlorophyll is present in these thylakoids. Thylakoids are arranged very closely in the form of stacks. So the thylakoids are arranged in the form of stacks. You can compare it with a stack of coins or a stack of books. These stacks are called grana. Now, is the chloroplast present in every cell of the leaf? No. It is present only in some cells of the leaf. For this, let's quickly go through the different layers of a leaf. There are several layers present in a leaf. See the image on the screen. It is the image of the section of leaf. A leaf contains the outermost layer of cuticle. It is a very thin and waxy layer, because of which leaves shine. Below the cuticle is the upper epidermis. It protects the leaf. The next layer is the palisade layer. After palisade layer comes the layer of spongy mesophyll. Now why this layer is called the spongy mesophyll? Because it has lot of air spaces in it. And the last layer is the lower epidermis. Out of all these layers, chloroplasts are present only in 2 layers, which are palisade layer and the layer of spongy mesophyll. Let's quickly see the procedure of this experiment which we can perform in the school laboratory. Take a potted plant with variegated leaves. What are variegated leaves? In such leaves, only some regions will have the chlorophyll present. So, the entire leaf will not be green. There will be some green patches on the leaf. The best example is the money plant. Its leaves are not totally green. You will find the green color in patches. Now, the second step of the experiment is keep the plant in the dark room for a few hours. Now, why this step is important? Keeping plant in the dark room will not give it any scope for photosynthesis. Hence, plant will consume all the starch present in its body. This is called de-starching. Removing the starch from the plant's body before the experiment is nothing but de-starching. The third step is keep the de-starched plant in sunlight for some hours so that photosynthesis will take place. After some hours, pluck a leaf from this plant and mark the outline of its green and non-green regions. Then perform a starch test on the plucked leaf. Keep the plucked leaf in boiling water for 1 minute. Now, dipping leaf in the boiling water kills all its cells. So, if any activity is taking place in the cells, it will be stopped. After that, immerse the leaf in the alcohol in the beaker and keep the beaker in a water bath. Heat the beaker till the alcohol boils. The significance of this step is alcohol removes all the chlorophyll present in the leaf. Then dip the leaf again in hot water. This is to remove the traces of alcohol present. Then place the leaf on the petri plate and add a few drops of iodine solution. What will you observe? You will see blue-black color in the green regions of the leaf, while non-green regions will not show any color change. Now, why is this so? Iodine reacts with the starch present in the green region and forms blue color iodine starch complex. Remember, only green region of the leaf showed the presence of starch. So, this indicates that the chlorophyll is important for photosynthesis. Now, let's assess yourself. Describe thylakoids, grana and stroma in one sentence each. Let's see the answer. Thylakoids are flattened sacs which contain chlorophyll. Grana are the stacks of many thylakoids arranged one on the other. Stroma is the colorless ground substance in which thylakoids, grana, DNA and ribosomes float. Now here remember, chloroplast is the organelle which has its own DNA. The second raw material is carbon dioxide. Carbon dioxide required for photosynthesis enters plants through stomata. What are stomata? Stomata are the minute pores present on the leaf surface. You will find these stomata present in the upper epidermis as well as in the lower epidermis. You can see the image of stomatal apparatus here. There are green color epidermal cells which surround the stoma. Now here stoma is singular and stomata is the plural word. You can also see two bean-shaped cells on both the sides of the stoma. Yes? These are called guard cells. Now the guard cells have a role to play here. The stomatal opening is not open always. In some part of the day it is open and rest of the day it remains closed. Hence, the gas exchange is not possible always. Or we can say the gas exchange is possible only when the stomata are open. Why does stomata open or close? The guard cells are responsible for this. The guard cells control the opening and closing of the stoma. Now compare this stomatal apparatus with a window. Assume the two doors of the window are the two guard cells. When water enters the guard cells, these cells swell, which opens the stoma, and hence the gas exchange is possible. Now when does the stoma close? When water leaves the guard cells shrink, which closes the stoma. Before we go ahead, let's take a small break.